Welcome back to another video of Considering Biology. For the chapter Biological Molecule, we have already covered one video in which we talked about the two groups of the biological molecule, that is the carbohydrate and the protein. In today's video for the Olevels Biology, we are going to cover the topic that is the nucleic acid. So nucleic acid and the lipids. So we first will talk about the nucleic acid. Basically, the nucleic acid is the organic compound which is found in the nucleus. So what you need to know about is that it is an organic compound and it is mostly found in the nucleus. It is found in the nucleus and it is made up of nucleotide. So number one pointer is that it is found in nucleus, and the other pointer says that it is made up of nucleotide. The nucleic acid is made up of a nucleotide, and by the term nucleotide, we mean that nucleic acid has the two major structures, that is the DNA and the RNA. Let's talk about the DNA and RNA in detail. The DNA stands for deoxyribonucleic acid, that is the deoxyribonucleic acid, whereas the RNA stands for the ribonucleic acid. DNA is basically a genetical material that helps in inheritance. It also helps to make the protein. Basically, it helps in the encoding of or the decoding of a protein. It is a genetic material. It is a genetic material and also helps in the coding of the genetic material that transfers from the one generation to another. Now, this is what you need to know about the DNA. Let's talk about the RNA. The RNA, what you need to know about the RNA is that RNA stands for ribonucleic acid. It does not perform a very important function when it comes to the metabolic and the catabolic activities that along takes place with the nucleic acid. All you need to know about the first three RNA. So DNA has a very quiet structure. It has a double helix structure. This is a very important pointer. It is also mentioned in your syllabus that how DNA differs from the RNA in the structure. DNA has a double helix structure. It consists of two strips made up of like the sugar and the phosphate twisted around each other and are connected by the nitrogenous bases. Now, what is the sugar, what is the phosphate, and what are the nitrogenous bases? Let's talk about them first, and then we'll come on to the point of double helix structure. Alright, so let's first talk about the parts of nucleotides. So nucleotide has three parts. It consists of a phosphate group. Then it has a base, and then it has a sugar molecule. These three constituents makes up together the nucleotide or the nuclear base. The phosphate group, the base, and the sugar. In the base, we have four kinds of the base. We have adenine, we have thymine, we have guanine, and we have the cytosine. Let's write them down. We have number one is the adenine, number two is the thymine, then we have guanine, and then we have cytosine. How are they paired along with each other is that adenine always comes in front of the thymine, whereas the guanine always comes in front of the cytosine. Thymine always alternatively comes in front of the adenine, whereas cytosine always comes in front of the guanine. Let's talk about them in more detail. This is how the pairing occurs in the nucleotide of the bases. Let's talk about it, how they are based and how they are linked together. The adenine and the thymine has a double bounded structure. The guanine and the cytosine has a triple bounded structure. Again, the thymine and adenine has a double bounded, and the cytosine and guanine has a triple bounded structure. These four are the conditions of the nucleotide structure that can be present and of the bases. And then we have adenine, we have thymine, we have guanine, and we have cytosine. Adenine and thymine are mostly opposite each other, and cytosine and guanine are similarly opposite of one another. Since in the last slide we were talking about how DNA is a double helix structure. Alright, so how it is, so in the double helix structure, these nitrogenous bases are linked together and are connected to each other by the group that includes the sugar, phosphate, and these bases. These bases include adenine, guanine, cytosine, and thymine. It looks like a spirally K-shaped structure. Let me draw it for you. This is how is the structure of adenine and guanine. On this structure, we have the bases. This is how the bases are attached, and they are linked together with a bond that is the hydrogen bond. Please do keep this in your mind. The bond that is present between the bases is known as the hydrogen bond. This is the double helix structure of the DNA, that is the deoxyribonucleic acid, which consists of a phosphate group, the base, and the sugar molecule. The sugar molecule is a ribose sugar molecule. This is some extra information that you might keep in your mind. That is, sugar molecule is a ribose molecule, whereas the phosphate group is a normal phosphate group. This is all you need to learn about the nucleic acid in detail. That we have learned about the DNA and the RNA, and we've also covered the parts of nucleotide. Alright, after the nucleic acids, let's discuss about the one last group, that is the fats. Fats are also known as the lipid group. So, fats are like the carbohydrates or the organic molecules which are made up of the elements that contain carbon, hydrogen, and the oxygen. But unlike the carbohydrate, fat contains much less oxygen in proportion. Let me give you an example. The testosterone is a beef fat, which is a very common example of lipids. The common formula for the testosterone or the beef fat is C57H110 and O6. Note that how the proportion of oxygen is so much less in number or in the proportion to the hydrogen. Hydrogen is around 110, oxygen is around 6. So the ratio of 106 oxygen is much more less and proportional than the hydrogen. So what does this mean? This means that the proportion of an element that makes up the fats are not fixed. So this is just a general formula of it. However, fats can be animal or it can be plants fats. So basically, there are two types of the fat. They can be animal and they can be plant fats. These are the two kind of fats that are available usually. However, fats can be broken down into fatty acids and the glycerol. What are the fatty acids and the glycerol? They can be broken down into, let's discuss how they can be broken down. They can be broken down into fatty acids and the glycerols by the process that is known as the hydrolysis. Hydrolysis is the kind of the process in which the water is involved in the reaction in order to form the product. Let me give you an example of the process of hydrolysis. Let me give you an example of the hydrolysis or the process of hydrolysis in the fat or the process of a fat. Suppose this is a fat molecule that involves three molecules of the water and gives us one glycerol molecule. This is a glycerol molecule and the three layers of the fatty acids. Fatty acids and glycerols are the minor products or the monomers that makes up the fats or the lipids themselves. So these are the three fatty acids. This is the glycerol, this is the water molecule, and this we have is the fat molecule. This is the process of hydrolysis in which water is involved in order to break down or in order to make up the product. That is, the fat molecule in the presence of water is broken down into glycerol and the three fatty acids. This involves the addition of water molecule and occurs very readily with an enzyme. For example, when a testosterone is hydrolyzed by the enzyme lipase or stearic acid and glycerol are produced. How this is so? For example, I've already told you about the testosterone, which is a very common fatty acid, and I've also told you about the its general formula in the last sheet. So, whenever the testosterone in the process of hydrolysis involves water in the presence of some enzyme, let's say the enzymes can be lipase or they can be stearic acid, but for this reaction we are taking the enzyme lipase. So when the testosterone involves the water molecule for the process of hydrolysis in the presence of the lipase, it produces glycerol and it produces stearic acid. This is a very common equation for the lipids when it comes to the hydrolysis of the lipids. All right. This is what you need to know about the how the testosterone or the how there is a production of glycerol and the fatty acids by the hydrolysis of the fats. So now let's talk about what are the sources of fat. The sources of fat include multiple products, or it actually includes the food that are rich in fats, include butter, cheese, fatty meals. Let's write them down, like say butter. Butter is a very common example of the fat. All the dairy products, cheese, or you can say fatty meat, olives, These are the very common sources of the fat. We have nuts, we have peas, we have beans, and like the meat of most of the fishes, let's say the white meat, is a very rich source of fat. It is a very rich source of fat. White meat is a very rich source of fat. However, in some fishes, such as herring or the salmon fish, have lots of fat in them also. But the white meat have relatively much more fat than the usual one. So, there's some small points that you need to learn about the topic fat. Let's discuss some of them. Number one is would be that what is the difference between the fats and the oil? This is mentioned in your syllabus. Fats and the oil. What is the difference between fats and the oil? So fats are basically, let's make a column that says fats are basically solid and oils are basically liquid at room temperature. At RTB stands for room temperature. Fats are solid and the oils are liquid at room temperature. So this is a very common difference that often come in your MCQs based questions. Let's say, what is the condition of a fat when it is at room temperature? So all you need to learn is that fats are solid at room temperature, whereas oils are liquid at room temperature. Another point that you need to remember is that lipids are the compounds that are soluble in the organic solvent. They are soluble in the organic solvents. Now, what kind of organic solvents? So, the kinds of organic solvents include the chloroform and the ethanol. Examples of liquid lipid include fat, waxes, and the steroid, however, but they are only soluble in the organic solvent. In organic solvent, we have chloroform and the ethanol. Now, these are some points that often come in your MCQ based question that what is the difference between fats and the oils. So all you need to remember is that fats are solid at room temperature and oils are liquid at room temperature. Another pointer says that they are only soluble in the organic solvent. In organic solvent, by organic solvent we include chloroform and ethanol in the organic solvent. These two pointers are very important and are also mentioned in your syllabus. Alright, at last let's discuss about the functions of fats. Fats have multiple functions, but the basic function is that if they are the source of energy for whatever the reaction is taking place, or even for the human body, the fats are very important for the source of energy. They're also known as for being an insulating material. And how so? It is so that they prevent excessive heat loss. They are the insulating material that prevents the excessive heat loss. Heat loss can be under the certain conditions, but excessive heat loss can be prevented by the fat. For example, animals such as seals that have a thick layer of the fat or bubbler beneath the surface, which helps to them to reduce the loss of body fat or the loss of body heat due to the fact they are stored into a very accumulating manner. And the third pointer says that a solvent for fat soluble vitamins. They are what? They are the solvent for fat soluble vitamins. They also act as a solvent for the fat soluble vitamins, an essential part of the protoplasm. They are the essential part of protoplasm. So these are the four main functions of the fat that says they are the source of the energy. They are the insulating material that helps to protect the excessive heat loss, and also they are the solvent for fat soluble vitamins. And they also have an essential plays an essential part in the protoplasm. These are the four pointers that need to learn about the fats. We have almost covered most of the topics of the biological molecule. We have talked about the carbohydrate, protein, lipids, and the fats, and also the nucleic acid. Now all we need to learn is the food test. In the next video, we will shall be learning about the food preparatory test. So that's it for the video. Thank you for watching.